Hello everyone, myself Dr. Mohini Kuchekar, Assistant Professor at PE Society's Modern College of Pharmacy, Nigri, Pune. Today I am going to present on topic Hybridoma technology. Learning outcomes. From the above topic, learner will be able to understand what is Hybridoma technology, its production method, its purification technique, and application of hybridoma technology, as well as the importance of monoclonal antibodies in the industries. So, an antibody is a protein used by the immune system to identify and neutralize foreign objects like bacteria and viruses. Each antibody recognizes a specific antigen unique to its target. Monoclonal antibodies are antibodies that are identical because they were produced by one type of immune cell, all clones of a single parent cell. Let's discuss the hybridoma technology. Hybridoma technology is a method for producing large number of identical antibodies, which is also called as monoclonal antibodies. This process starts by injecting a mouse with an antigen that provokes an immune response. A type of white blood cell, that is B cell, will be converted into plasma cell by the presence of antigen. The plasma cell will then produce an antibodies that binds to the antigen are then harvested from the mouse. This isolated plasma cell are in turn fused with immortal B cell cancer cells, which is nothing but the myeloma cell, to produce the hybrid cell line, and that hybrid cell line we called it as a hybridoma. In short, if we'll discuss the hybridoma technology, it is a technique of producing hybrid cell lines called hybridomas by fusion of specific antibody producing lymphocyte B cell with the myeloma cell that has an ability to grow in tissue culture. Hybridoma produce antibodies that have single specificity and are called as monoclonal antibodies. Basically, this technique was discovered by scientists Colette of West Germany, Milstein of Argentina, and Journey of Denmark in 1975. These scientists were also awarded as a Nobel Prize for Physiology and Medicine in the year 1984. Let's discuss the actual steps which involved in production of the monoclonal antibodies. That is, the steps which are in the hybridoma technology are follows. First step, that is the immunization of specific animal. Second step is isolation of myeloma cell. Third step is fusion of the spleen cell and myeloma cell. Fourth stage is selection of HAT medium. Fifth step is isolation of hybridoma cell, and sixth step is screening of this isolated hybridoma cell. Seventh step is in vitro or in vivo multiplication, and the last step, which is nothing but and very important step, that is the purification of antibodies. Let's discuss with the first step. So as I discussed earlier, the first step, which is immunization of specific animal. So as we have already discussed, we generally use the mice. So an antigen is immunized to an animal via intravenously by the injection, where in the spleen it activates B cell which produce plasma cell. These plasma cell to produce the monoclonal antibodies, and then the isolation of this plasma cell from the spleen of animal is carried out. Second step, that is the isolation of myeloma cell. In the isolation of myeloma cell, the myeloma cells are the cancerous cell which is isolated from bone marrow. These myeloma cells are generally immortal in nature and has multiplication property. After that, this third step, that is fusion, which is very important step. Here in this step we are going to fuse the spleen cell and myeloma cell. For the fusion of the spleen cell and myeloma cell, we need the chemical which is a PEG, which is nothing but polyethylene glycol. It can also done by another technique which is called as a electroporation technique or electrofusion technique. By applying an electric field, we can go for the fusion of spleen cell and myeloma cell. So fusion between spleen cell and myeloma cell produced 5 different types of the cell. It is not like that in the very first step directly the fusion will gives you the hybridoma. There are various chances are there, and depending upon that different types of the cells are produced. That means here we will discuss it one by one. That is, we can get a fused plasma cell, we can get fused myeloma cells only. Either we will get hybridoma, unfused plasma or unfused myeloma. The next step is selection of HAT medium. So HAT is nothing but hypoxanthine aminopterin thymidine. So before multiplication of antibody, it has to synthesize a new copy of DNA, and for that it requires synthesis of nucleotide. The synthesis of nucleotide mainly it occurs by two pathways. So first pathway is salvage pathway and another pathway is de novo synthesis. So let's discuss the salvage pathway. So this salvage pathway, it requires degraded part of old nucleotide to produce new nucleotide. And in the second de novo synthesis, it synthesized completely a new nucleotide by a small molecule. So in HAT medium, cells not synthesized by de novo synthesis due to the presence of aminopterin in HAT medium, because it blocks dehydrofolate enzyme, which is very necessary for the synthesis. And that's the reason for the synthesis in salvage pathway, it must require HGPRT enzyme. HGPRT is nothing but hypoxanthine guanine phosphoribosyl transferase, where hypoxanthine and thymidine are used as precursors. Next step, isolation of hybridoma cell. Here, as after the fusion, the spleen cell have HGPRT enzyme and myeloma cell it doesn't have HGPRT enzyme. So after the fusion, we are mainly interested for the isolation of hybridoma cell. So here the 5 types of cells are produced like fused plasma, fused myeloma, hybridoma, unfused plasma, unfused myeloma, where only in fused plasma, hybridoma and unfused plasma HGPRT is present. Here we will get a better picture. Here the spleen cells are there which is having and HGPRT antibody. Here, the myeloma cells which doesn't have HGPRT, we are going to fuse it with the polyethylene glycol, and after fusing, it is going to produce unfused spleen cell, fused spleen cells, most important in which we are interested, that is hybridomas, then fused myeloma cell and unfused myeloma cells. Out of that, we are going to take the hybridoma cell for further proliferation. Fused myeloma and unfused myeloma doesn't have HGPRT enzyme, as we discussed, so can't survive in heart medium. The fused plasma and unfused plasma have HGPRT enzyme but didn't have a long life. Hybrid cell has HGPRT enzyme from the spleen cell, as well as they have the ability to multiply repeatedly as myeloma cell. So the isolation of hybrid cell because is only cell which survives in the heart medium. Then sixth step, which is nothing but the screening. So in the screening, ELISA screening method which done by incubating hybridoma culture, in which secondary enzymes get conjugated and formation of color product shows positive hybridoma, which is used for multiplying the hybridoma cell. In vitro method, it involves culturing of hybridoma cell in a suitable culture media, and then antibodies are isolated and further purified. The next, in vivo. So in vivo procedure, it basically involves introduction of hybridoma cell into the peritoneal cavity of animal. Then from the ascitic fluid, the antibodies are isolated. Once the hybridoma colonies is established, it will continually grow in culture medium like RPMI 1640 and produce the antibodies. Here, for the storage, we prefer the liquid nitrogen. The next and very important step, which is nothing but purification of antibodies. So, in the purification of antibodies, monoclonal antibodies may need to be purified before they are used for variety of purposes. So for the purification, we generally follow the chromatographic technique like ion exchange chromatography and antigen affinity chromatography. Serum-free media for the bulk culture of hybridoma cell is very important. The use of serum, however, it leads to difficulties in the purification process of the antibodies. It is an expensive technology for large-scale production of hybridoma cell for industrial production of monoclonal antibodies. In the view of this difficulty, serum-free media are being increasingly used for culturing hybridoma cell. So this is the outline of steps of hybridoma technology which we have already discussed one by one. Then let's discuss the advantages of serum-free media in hybridoma cell culture and preparation of monoclonal antibodies. Greatly simplified purification of antibodies due to increased initial purity and absence of contaminating immunoglobulin. Decreased variability of culture medium. It reduces the risk of infectious agent. Fewer variables for quality control and quality assurance are required. Increased control over bioreactor conditions. Potential for increased antibody secretion. Low or no dependence on animal. It is very cost effective method and overall it increases the efficiency. Then let's discuss the disadvantage of serum free media in hybridoma cell culture and preparation of monoclonal antibodies. Not all serum free media are applicable to cell lines. Cell may not grow to as high densities and may be more fragile than the cell in serum. Media may take longer to prepare. Hybridoma technology is a laborious and time consuming process. This is no guarantee that monoclonal antibody produced is totally virus free despite the purification. For this reason, US Food and Drug Administration insists that monoclonal antibodies for the human use should be totally free from all pathogenic organisms including viruses. The last point and very important, which is nothing but its application. So it has wide applications, that is, it helps in identification of ABO blood group, in the detection of pregnancy, biochemical analysis in diagnosis of pregnancy, cancer, hormonal disorders, and infectious diseases, purification of individual interferons using monoclonal antibodies, inactivation of T lymphocyte responsible for rejection of organ transplant, removal of tumor cell from bone marrow. It is used in the treatment of acute renal failure, treatment of malignant leukemic cell, B cell lymphomas, and variety of allograft rejection after transplantation, and various industrial applications. In concluding, hybridoma technology is the valuable for preparing antibody in vitro condition, monoclonal antibodies, as they are known to have an open, remarkable new approach to preventing, diagnosing, and treating disease. It is helpful in various aspects. So we have discussed the various points in this. That is, what is hybridoma technology, the production and purification technique, advantages and disadvantages, along with its industrial application, we have covered in this topic. These are few references which I have gone through. Thank you.